Okay, well, let's jump into some examples and you lay some out in your coverage of this particular topic. The first is something called two-component regulatory systems. Now, what are these systems? What are the two components and what general purpose do they serve for bacteria?
Yeah, so bacterial cells use two-component systems to sense and respond to changes in the environment, and we find those throughout bacterial organisms. It is very, very common in the prokaryotic world. And as their name implies, two-component systems essentially have two major components. You have the sensor histidine kinase and a response regulator. And the sensor kinase, in response to chemical cues, undergoes autophosphorylation, which means that a phosphate group is transferred from ATP to a histidine residue on the kinase. And the histidine kinase has itself two domains. So it has an input domain and a transmitter domain. And the input domain is located on the cell's exterior, so outside of the cell, and it's particularly situated to detect incoming environmental signals. And the the transmitter domain is situated on the cytoplasmic face of the cell membrane, and so it's positioned so that it can interact with the response regulator. So the phosphate group that remember I mentioned that the histidine residue of the response of the histidine kinase gets a phosphate group transferred to it from ATP, and then this phosphate group in turn is transferred to the response regulator. So that's the second component of the two-component system, which then drives a cellular output, such as turning genes on and off, etc. And so, as I mentioned before, two-component regulatory systems are extremely common among bacteria, and each utilizes the same basic design logic, even though these are not evolutionarily descended from one another.
Okay. All right, that's a good basic breakdown of it. So you've got a sense and response system that is common across different organisms, even wildly different organisms, as you've pointed out. Now, you give the example of E. coli regulating outer membrane proteins in response to environmental osmolarity using a two-component system like you're describing. How does this system allow the bacterium to sense changes in its surroundings and then respond appropriately?
Yeah, so osmolarity is essentially a measure of the concentration of solute particles in a solution. And the cell needs to regulate the expression of porins in response to this environmental osmoregularity. And it does so by means of a two-component system. So in this case, the sensor kinase is is a protein called MZ, and it's located in the inner membrane. And MZ detects osmolarity changes, and it undergoes autophosphorylation. And then the response regulator for the system is known as OMP-R, and it receives the phosphate group from MZ, and it regulates the expression of genes. So when osmolarity is high, the kinase activity of MZ is activated, and this results in the phosphorylation of OMP-R. And when osmolarity is low, the phosphatase activity of MZ is activated, and this reduces the levels of phosphorylated OMP-R. So when OMP-R is phosphorylated, it becomes an active dimer that has enhanced DNA binding ability that's specific to OMP-C and OMP-F gene promoters. These are porin genes that encode outer membrane proteins, which allow the passage of metabolites across the outer membrane of gram-negative bacteria. Gram-negative bacteria are bacterial cells that have two membranes, an inner and outer membrane. So the pore diameter of OMP-F is larger than the pore diameter of OMP-C. So this allows for a tenfold faster diffusion rate, which is advantageous under conditions of low osmolarity, where there are scarce nutrients. If osmotic pressure is low, the synthesis of OMP-F is increased, whereas if the osmotic pressure is high, the expression of OMP-C is increased. And also the transcription of McF antisense RNA is initiated, and this essentially blocks the transition of OMP-F by complementary binding, and that represses the synthesis of OMP-F. So this is controlled by a two-component system, making use of a sensor kinase as well as a response regulator.
Okay. Yeah, and that's that's a good dose of technicality, as it were. And I always hope that the people that are listening or watching are not going to get scared away by the technical details. As Michael Behe challenges us in Darwin's Black Box, we've got to bite the bullet of complexity if we want to understand why Darwinian evolution is not capable of this. So, audience, stay with us. This, we really do need to get into the technical aspects of this a little bit just to understand why. So Jonathan, I can always depend on you for that, and I'm grateful for that. Okay, so another example that you've laid out is something called chemotaxis, and that's quite a fascinating example in itself, especially the idea that bacteria have kind of a memory system for chemical concentrations. Can you explain how that system works and how it helps bacteria navigate their environment?
Sure. So chemotaxis is really a signal transduction system that enables the bacterial cell to move towards attractants like glucose or away from poisons. And it does so by modulating the frequency of runs and tumbles. So basically, the flagella of the bacterial cell rotate as a bundle, and their default is to rotate in a counterclockwise way. But in response to chemical stimulus, it can switch direction and start spinning clockwise. And this results in the bacterial flagellar bundle breaking apart, and that results in the bacterial cell tumbling. And if you are moving towards a food source like glucose, then you want long runs and less frequent tumbles so that you can move in the direction of that food source. Whereas if you're moving towards a poison or you're moving away from a food source, then you want to have frequent tumbles and shorter runs so that you can reorient yourself and sample the environment and try to get closer to that food source or away from the poison. And the chemotaxis system is, again, a two-component system. So in this case, so you have embedded in the cell membrane methyl-accepting chemotaxis proteins, and different methyl-accepting chemotaxis proteins, which are also known as MCPs, can detect different types of molecules, and they're able to bind attractants or repellents. And these receptors then communicate with and activate the so-called key proteins. And so there are proteins called key A and key W, which are bound to the receptor. Key W essentially functions as an adapter protein, but key A is what we call the sensor kinase or histidine kinase for this system. And upon activation of the receptor, the conserved histidine residue of key A undergoes autophosphorylation by ATP, so it obtains a phosphate group. And there are actually two response regulators called key B and key Y. And there's a transfer of a phosphoryl group to the conserved aspartate residue from key A, right? So key B and key Y both receive a phosphate group from key A, and then the key Y protein is subsequently interacts with the flagellar switch protein that's known as Fly-M, and that induces the switching in flagellar direction from counterclockwise to clockwise. And so this is, again, a two-component system, which is analogous to the system that we reviewed previously regarding osmoregulation.
Yeah.